Okay, so welcome back to this next video in which we are discussing the Western blot. Okay, so we have now got a copy of our array of proteins on a nitrocellulose or PVDF membrane. Okay, and now what we have done is we have blocked all other proteins being able to bind to this membrane arbitrarily by dousing the membrane in bovine serum albumin, which is a protein. Okay, so now nothing can bind to the membrane unless it actually binds to one of the proteins that is already bound to the membrane. Okay, so now what we will do is we will expose our membrane to the primary antibody. Okay, and this is our anti-protein A antibody. So remember, protein A was our protein of interest. We want to see whether protein A is expressed within the cells that we lysed to get the cell lysis. Okay, so we will now expose our nitrocellulose or PVDF membrane to the primary antibody here. And basically, incubation with the primary antibody generally is done overnight, okay? You can do it in around an hour, but the results won't be as good as if you do it overnight. So usually, it's incubated overnight in a cold room. Okay, so what will then have happened is the primary antibody will bind to the denatured protein A, okay, but it won't bind to any of the other proteins, okay, that are also bound to the nitrocellulose or PVDF membrane. Okay, so now all over the membrane where you've got protein A bound, you will also have another antibody bound off that. Okay, right. Then what you're going to do is, after overnight incubation with the primary antibody, you'll then take your nitrocellulose or PVDF membrane out again, and you'll wash off the remaining primary antibody that hasn't found a protein A to bind to, and then what you'll do is you'll incubate it with the secondary antibody, and this generally isn't done overnight. This can be done in an hour, basically. So what you'll then get is an antibody which binds to the primary antibody, basically. So antibodies have two main regions, so let me highlight the two different regions. So highlighted in blue here, these are the antigen binding portions of the antibody, okay, often denoted FAB for antigen binding fragment, or the fragment antigen binding. And then the rest of it, basically, is then what's known as the FC, which stands for the fragment crystallizable. Now basically, the primary antibody, which is against protein A, will have been raised in some animal, basically, potentially a rabbit, okay? So you will have made an antibody in the rabbit against the denatured protein A. And that's where this primary antibody has come from. Then what the secondary antibody will be is it will be raised in a totally different animal, basically, maybe a donkey, let's say, okay? And it will be raised against antibodies that are produced in rabbits, and it will bind to the FC portion of rabbit antibody, basically. So maybe this will come from donkey. So basically what you will then have is these secondary antibody molecules which will bind to the crystallizable fragment of all rabbit antibodies, basically. So now what you'll do is expose your PVDF membrane to the secondary antibody. The secondary antibody will now bind to the primary antibody, like so, okay? So where you had your protein A on the membrane, so these strips here, you will now have the anti-protein A antibody, the primary antibody bound there, and then on top of the primary antibody you'll now have a secondary antibody bound on top of there. Okay? And multiple secondary antibodies can bind to the same primary antibody, because these aren't necessarily monoclonal antibodies against the primary antibody, okay? So let me just explain what the difference between monoclonal antibody and polyclonal antibody is. So what you have to understand these terms is that people use the term antibody to mean basically a flask of fluid that contains absolutely loads of antibody molecules. You have to understand that some people don't use the word antibody to mean an antibody molecule. They mean a solution containing absolutely loads of antibody molecules. And the terms monoclonal antibody and polyclonal antibody refer to the use of the word antibody to mean a whole solution of loads of antibody molecules. So if people refer to monoclonal antibody, what they mean is that they have a solution of antibodies which bind to, let's say, the rabbit fragment crystallizable of the antibody produced in rabbits, okay? And they are all absolutely identical. So if you've got a monoclonal antibody, it means all of the antibody molecules in here are identical. They all have identical antigen binding regions. They all bind to the exact same portion of the fragment crystallizable, basically, of the primary antibody. Okay, so basically, if you use a monoclonal antibody, only one secondary antibody will be able to bind to the Fc region, because after one has bound, then the specific part of the Fc region which the antibody binds to will be occupied, and therefore you won't be able to bind another one on there. So the concept of the portion of a protein which an antibody binds to is known as an epitope. So basically, monoclonal antibodies all bind to the same epitope of a protein. If you have a polyclonal antibody, what that means is that you have a solution containing loads of antibody molecules, and all of the antibody molecules are targeted against a single protein, so they all bind to a single protein. However, they don't all bind to the same epitope, so they might bind to different little domains of the protein, or the different epitopes. Okay, so you can get multiple antibody molecules binding to the fragment crystallizable of the primary antibody if we are using a polyclonal antibody against rabbit antibody, basically. Okay, and indeed we probably will be. Now, firstly, the advantage to using a secondary antibody is that you can massively scale up the signal, basically, because one primary antibody can get a huge number of secondary antibodies bound to it. The other advantage is that we are about to tag these antibodies with a enzyme, basically, known as horseradish peroxidase. Now, tagging enzymes onto antibodies is an expensive and time-consuming process. Okay, so would you want to go through every possible primary antibody against every different protein and tag them all with an enzyme? Okay, the answer is no. Remember, the primary antibody, protein A is arbitrary. The protein that we're interested in is arbitrary. Different researchers around the globe will all be looking at different proteins, basically, so they will all be working with different primary antibodies. If we tagged the primary antibody, then for every different primary antibody, we would have to conjugate enzymes to all of those different primary antibodies. That would be very expensive. The advantage of using a secondary antibody is that this secondary antibody will bind to loads of different primary antibodies. It just binds to any old antibody that's come from a rabbit, basically. Okay, so you can make loads and loads of different primary antibodies that are all from rabbit, and the secondary antibody will bind to them all. This means that you can make one secondary antibody which has an enzyme conjugated to it, and loads of researchers all around the world can use that same conjugated antibody. You don't have to make different conjugated antibodies for every different researcher. Okay, so that's another advantage of using secondary antibodies. So let's now talk about the fact that we've conjugated these secondary antibodies with a protein, okay, specifically an enzyme. So we've now attached an enzyme onto all of these secondary antibodies, okay, shown here in blue. Now this enzyme is known as horseradish peroxidase. Okay, so this is horseradish peroxidase, and for short, horseradish peroxidase is often abbreviated to HRP. Okay, so basically, let's go over where we've got to now. looking at this nitrocellulose PVDF membrane again, we have our protein A strips. What will have happened is that the primary antibody, which was anti-protein A antibody reared in rabbits, will bind there and nowhere else, because we've blocked the rest of the membrane, so it won't just bind to the membrane because it's just binding to the membrane. It will only bind to the membrane because it's binding to protein A and that is bound to the membrane. Then what will happen is we will put secondary antibody onto this membrane. Okay, this will bind to the primary antibody. Okay, so now we'll get secondary antibody all over these 4 lines. Okay, and this secondary antibody will be conjugated with an enzyme, so it will have an enzyme attached to it. So now we have horseradish peroxidase here, 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 and here. Now we're going to be able to visualize the presence of horseradish peroxidase. Now, how are we going to do this? Well, basically, we're going to use a technique known as chemiluminescence. Okay, so this is chemiluminescence. Now, chemiluminescence is a very general technique. It basically means a series of chemical reactions which release light. So we are going to be able to set up a series of chemical reactions that will be catalyzed by the horseradish peroxidase and which will result in the production of light. Hence, what will happen is we will get light being emitted from these 4 lines, and we'll be able to take a picture of that. Okay, so let's now talk about the reaction that's going to occur. So basically, what's going to happen is you're going to start off with a molecule known as luminol. So let me show you the structure of luminol. Okay, so you start off with a benzene ring here, so let's have alternating double and single bonds, and this is going to be luminol. Okay, then you have another ring attached off this one here, like so. Okay, and you'll have an amino group coming off up here. Then you'll have 2 carbonyl groups, one here and one here. And I've forgotten to put in the nitrogens, and I'll do this repeatedly when drawing this structure. I always have a weakness for drawing this structure. There should be a nitrogen here and a nitrogen here. So we're drawing a skeletal structure. So most of these are carbons, but these 2 here are nitrogens, and then they also have hydrogens coming off them, like so. Okay, so I'm sorry for that mess. I'll try and remember to draw the nitrogens in the next step. Okay, so this is luminol. Now, what we're going to do is we're going to put luminol in a solution where it has high hydroxide anion concentration. Okay, so we're going to need 2 hydroxide anions, basically. And what's going to happen is these hydroxide anions are going to nick these hydrogens from the luminol molecule. So, we're actually going to draw an electronic flow diagram for this, joy of joys. Okay, so I'm going to actually draw what a chemist would draw. I'm going to show you where the electrons will move. So, the 2 electrons in this bond will go back to the nitrogen. Now, one belonged to the nitrogen, so that doesn't matter, but the other one belongs to this hydrogen, so that means that this nitrogen is gaining an extra electron, and therefore is going to become negatively charged. The same thing will happen here. The hydrogen atoms have lost their electrons, so they gain a positive charge, and what they will do is they'll go and bind with these 2 hydroxide anions to produce 2 water molecules. Okay, because they don't need electrons, because the hydroxide anions have already got an extra electron which they can give to the hydrogen atom effectively. And what you will produce from this is shown here. Okay, so here we still have our benzene ring with the alternating double and single bonds. We have our amino group here. Okay, and then off this, like so, and I'll try to remember the nitrogen this time. We now have these nitrogens which have negative charges, so they're both negatively charged, and then coming off this carbon you have a carbonyl, and coming off this carbon you have a carbonyl as well. Okay, right. Now, what's going to happen next? Well, basically, what's going to happen is these nitrogens, which have these extra electrons, are going to give these extra electrons into bonds, basically. So these nitrogens have got a lone pair of electrons that they shouldn't have. They're actually going to now have 2 lone pairs of electrons. But basically, they gained both of the electrons that were in this bond. So they've got this extra lone pair of electrons. They're going to provide both electrons, basically, into a new bond that they will form with this carbon atom. So this nitrogen will form an extra bond with this carbon atom, and this nitrogen will form an extra bond with this carbon atom. So both of these electrons are going to be given by the nitrogen, and it's basically going to give one to the carbon and say, hey, let's make another bond. So we're going to turn these two into double bonds. Now you will notice a problem there. These carbon atoms are going to end up with 5 bonds if that actually happens. So what they're going to do is they're going to cleave one of these bonds that is in this double bond between the carbon atom and the oxygen atom. So both electrons in one of these bonds will end up going back to the oxygen, okay, and the same over here. So the carbon doesn't mind that, because basically it gained an electron from this nitrogen when the nitrogen put 2 electrons into another bond between this nitrogen and this carbon. Basically, the understanding of a covalent bond is that 1 electron is from each of the 2 members. So basically, it's as though the nitrogen gave one of those electrons to the carbon and then said, right, let's form a double bond. Let's form an extra bond between us. Okay, I've got this electron and you've got that electron that I just gave you. Let's share them. Okay, but then the carbon atom has gained an extra electron, so it doesn't mind that it's losing an electron by both of the electrons in one of these bonds going to the oxygen. Okay, so what will you then produce? Well, here is the benzene ring. Okay, and so far we haven't seen the step that is actually catalyzed by horseradish peroxidase. We will do. At the moment, this is the step that is happening just before you add just before you add it onto the nitrocellulose membrane. So what will happen is you'll get like a kit, and one will be the luminol molecules, and the other will be like a solution that you mix with the luminol molecules, and this will contain the hydroxide anions, or the source of hydroxide anions. So you mix them together, and then everything can start. Then you tip the solution onto your nitrocellulose membrane, and the final step in the reaction is going to then be catalyzed by the horseradish peroxidase, and that's the step that then releases the light. So we'll get to that in a moment. Okay, so the next step, and I've nearly forgotten these nitrogens again, never mind, saved it just in time. So we've now got these oxygens with negative charge because they've gained an extra electron by taking both of the electrons in one of these bonds. Okay, we've got double bonds here and here, and we've still got a single bond there. Okay, so the next step is that an oxygen molecule is going to come in. Okay, so in comes our oxygen molecule, like so. So the next thing that you need to add in is this oxygen molecule. What's going to happen is, where shall I start with this reaction? Okay, so we'll start off with this bond here breaking. So basically we're going to break this double bond here, okay? And what we'll do is we'll give both electrons in that back to this nitrogen here, but then it will give one of these electrons to this nitrogen. So basically the 2 electrons that are in that second of the 2 bonds there are going to go into a new bond between this nitrogen and this nitrogen. Now of course that's going to leave this nitrogen with 1 too many electrons and this carbon with 1 too few electrons. So now what's going to happen is this bond here, this second of the 2 bonds there, is also going to break. Okay, and the 2 electrons in here are going to go into forming a bond between this carbon here and this oxygen here. So both electrons basically are going to be given back to this carbon, and it's going to say, hey, I'd like to make a bond with this oxygen. Now of course that leaves a problem, because now this oxygen has 1 too many electrons. So what will then happen is this bond here will break, and it will go into creating a bond between this oxygen and this carbon. So remember, this carbon had 1 too few electrons right from the first reaction. So we basically have this great cycle here. Now, what's the overall result of that? Well, you're going to produce a peroxide bond right through the middle of that 6-membered ring over there. Okay? So here's our, whoops, and I've forgotten the amino group. Here's the amino group. Okay? And there's an amino group here. Okay, and then we've got this other 6-membered ring here, and the nitrogens are still there, like so. They've now got a double bond between them, because remember, the 2 electrons here went into another bond between the nitrogens. This carbon is now attached to an oxygen. you've got a peroxide link between the two oxygens, because remember, they no longer have a double bond, and then it's linked to that carbon there, and then both of these have single bound oxygens to them, which have negative charges. Okay, now all of this happens without horseradish peroxidase. So when you mix the luminol with its reagent, then you'll get this stuff being formed. Now, horseradish peroxidase is going to react with, it's going to catalyze the reaction of this, which will release light. So basically, what you've done is you've mixed them together. Now what you'll do is you'll get your membrane, okay, you'll put it in a machine which is going to detect the light being emitted from this reaction that's about to occur. Then you'll coat the membrane in the mixture of the luminol with its reagent, which will now have these molecules in, and then once it comes into contact with the horseradish peroxidase, this next reaction will occur and light will be given off, and the machine will detect it and take a picture of it for you. Okay, so what will you end up with then? Okay, oh, well actually I need to tell you the next reaction before I tell you what you're going to end up with. Okay, so horseradish peroxidase, it breaks peroxide links. Peroxide links are when you have oxygen linked to another oxygen by a single bond, i.e. this is a peroxide link. So it's going to break that bond between those two oxygens. Now how is it going to break it? Well basically, what's going to happen is both of the electrons in this bond are going to go back to this oxygen atom here. Okay, so you're going to break that bond and give both electrons to that oxygen atom, so that that oxygen atom is going to end up with a negative charge. Okay, then that leaves, well, not yet. What's then going to happen is that both electrons in this bond here are going to go into a bond, an extra bond between these two nitrogen atoms. Okay, so you're going to take both electrons here and they're going to go to make a new bond between this nitrogen and this nitrogen to turn this into a triple bond. That leaves this nitrogen with too many electrons, so what's going to happen? This bond is going to break. And both of the electrons in this bond are going to go into this oxygen over here. Okay, to give that oxygen an extra electron, because remember it's lost an electron. So let me just revert back a bit. This, you might say, well, why on earth do both of the electrons here go to this oxygen? That oxygen wasn't even involved in this bond, but they do. Both electrons go to this oxygen, and you might say, well, doesn't it now have a -2 charge? Well, no, because it already had a +1 charge because it had lost an electron when this oxygen over here selfishly nicked both of those electrons. So it only gains a -1 charge. Now, what does that leave us with? Well, what's firstly going to happen is this nitrogen atom is going to be given off, well, nitrogen molecule rather. So nitrogen triple linked to another nitrogen is just what we would call nitrogen gas, and it makes up 78% or something of the atmosphere. Okay, now what does this leave us with? What problems does this leave us with? Well, this carbon here and this carbon here have both got too few electrons, so they've both got a positive charge. So what is of course going to happen is that these oxygens here are going to give their lone pair of electrons, which is giving them a negative charge, back in. So it will give 2 electrons, one will obviously stay with the oxygen and the other will go to the carbon. That will neutralize this oxygen and this carbon, and you'll create a double bond there. So what will you end up with finally? Let's draw this out. So here you have this benzene ring with the amino group here, like so, and then off it you then have now an oxygen atom here, okay? So I'm now drawing these oxygens that are linked to these carbons in the place where I previously showed the nitrogens, because they've gone now. Okay, and then we have carbonyl groups here, like so. And I think that is everything, apart from the negative charges on these oxygens. Okay, now, when this reaction occurs, not only do you give off nitrogen, but you also give off light, basically. So you give off energy in the form of electromagnetic radiation. Okay, the apparatus that you've put your membrane into will then take a picture of this light basically for you, and you will get a signal, and you will be able to see where your bands corresponding to protein A are, and you'll be able to see whether protein A is therefore expressed within the cell which you lyse to get your cell lysate. And it is this final step here that is catalyzed by horseradish peroxidase. Okay, so the only thing now to say is, why did we do 4 separate samples of different concentrations? Well, basically, what we would want to see is that the signal varies in accordance with that, okay? Because if we want to get an idea of quantity, we want to make sure that our reaction is not saturated. Okay, so what do I mean by this? Basically, if we think about what we had in sample 1, 2, 3, and 4, sample 1 had some concentration that we don't know, basically, but we're calling that 1. Then sample 2 was a half that concentration, sample 3 was a fifth that concentration, and sample 4 was a tenth that concentration. Okay, so what we will want to see is the actual amount of light being emitted from each of these bands, basically. We will want to see that the amount of light being emitted from, say, band A for sample 2 is a half as much light as was being emitted from band 1. We'll then want to see that the amount of light being emitted from protein A's band in sample 3 was a fifth of the amount of light that was being emitted by band A in sample 1. And we'll want to see that the amount of light being emitted in sample 4 is a tenth of the amount of light as was being emitted in sample 1. We want to see a nice linear relation, because that shows us that the amount of light that's being emitted basically is proportional to the amount of protein that's actually present. Now, you might say, well, why wouldn't it be? Well, basically what could happen is the reaction could get saturated, basically. For instance, let's imagine that we put in too little protein A. Put in too little anti-protein A antibody. So let me try and explain what the purpose of this is for. So imagine that you were being really tight on your budget, okay, because antibodies are extremely expensive, and you put in a tiny little amount of anti-protein A antibody, okay? Now, if we had let me get the picture back again. So if we had our nitrocellulose membrane here, we know that in these 4 different samples, there's different amounts of protein A. So in sample 1, we would expect there to be 10 times more protein A than in sample 4, for instance. Okay, but if we put in a ridiculously tiny amount of anti-protein A antibody, then, there might be less antibody than there is protein A, even in sample 4. So basically, let's say, just to put some numbers on here, because actually making it into an actual example is easier than trying to do it in general. So let's say there are 100 protein A molecules in this band for sample 4, okay, just to give numbers. That would mean that there is then 1,000 or thereabouts in the one for sample 1, 500 in the one for sample 2 here, and then 200 for the one for sample 3 here. Okay, like so. Imagine that we only put 50 protein A and anti-protein A antibodies on. Then 50 would bind here. Well, Imagine that we didn't put enough antibody on. Imagine that we did only put, let's say, rather than 50, let's say we put 200. Then what could happen is 50 could bind to each of them. Okay, so 50 would bind here, 50 would bind here, 50 would bind here, 50 would bind here, and then the secondary antibody would be put on, and the secondary antibody detects how much anti-protein A antibody there is, but now there's an equal amount of anti-protein A antibody for each of them. So actually the signal that you get for each of them would then end up being the same. So if you were too mean on the anti-protein A antibody, this illustrates how it could go horribly wrong, basically. How you might not end up with that linear proportionality, basically, where the signal increases in tune proportionally to how much protein there is, basically. Okay, so that's why we'd want to make sure that the common sense relationship that you see is actually what you would see, so that you actually can be sure that if, for instance, you do an experiment where you want to see that protein A has gone down, so let's say you do something to the cell, okay, that you think should make the expression level of protein A go down, let's say. And what you might want to do is you might want to use the signal that you see on a Western blot to show that the amount of protein A within the cytoplasm has gone down. Okay, but if there's something silly like this going on where it's already saturated, you might not actually see it go down even though it has gone down, basically. So you do want to make sure that this common sense relationship occurs, especially if you're going to be relying on the signal of the band to tell you how much protein A you have expressed within the cells. Okay, and that concludes our discussion of the Western blot.